So let's talk for a second about our procedure 5 from lab 5. So with this experiment, we were looking at the activation of amylase. That's the enzyme that breaks down starch into reducing sugars. So we had 8 different test tubes that were tested here. Our first 2 were just water. We tested test tube 1 with iodine and 2 with Benedict's. So if you remember our tests from lab 4, we had a reaction where test tube 1 was negative for starch and test tube 2 was negative for our reducing sugars because water, right? Water. Okay. Now then we tested test tubes 3 and 4 with starch. So iodine tested with starch and Benedict's tested with starch. Test tube 3 is one of our positive controls here because what happens, starch and iodine positive, but starch and Benedict's negative. 5 and 6, we tested reducing sugars for iodine and then for Benedict's. So we're basically just making the same test tube, but we're testing it with 2 different things, which we can't do in the same test tube, so we had to make 2. All right, so here we are at test tube 5 and 6. Woo, bad. All right, so we were negative for starch when we had test tube 5, but we were positive for reducing sugars. And this test tube is definitely sunk a little bit, but it's definitely positive in that orangey green color for reducing sugars. So those were all kind of controlees, right? So we had negative controls for test tubes 1 and 2 where we looked at water, positive control and we tested starch with iodine, and then a positive control and we tested reducing sugars with Benedict's. We were also testing to see that if we let things sit for 20 minutes that it didn't change, right? Starch remained starch after 15 or 20 minutes. Reducing sugars remained reducing sugars, didn't form any sugar or starch at the end. But with test tubes 7 and 8, we gave the enzyme starch, and then after 15-20 minutes, we tested it with iodine and then Benedict's. So it's the same thing, we're just doing 2 different tests. So what happened here is we had a negative test for iodine, which means there was no starch left. So we started with starch and amylase with number 7, and we ended with none. No starch was left. Now then when we look at the Benedict's solution, which has gotten a little weird after it's been sitting here for a little bit, starch and amylase produced sugar, which was positive for our Benedict's solution. So our Benedict's solution showed here that we have reducing sugars at the end of this test, even though we started with starch, which means that if we started with starch, we mixed it with amylase. We had no starch left at the end, but we had reducing sugars at the end. So we saw a breakdown of starch, aka a hydrolysis reaction, for the use of amylase.